Okay, so in the first video we introduced metabolism and we talked about digestion, where polysaccharides are broken down into monosaccharides. This begins in the mouth with salivary amylase and is completed in the small intestine with pancreatic amylase, where these individual monosaccharides, or I should say the individual monosaccharides here, are absorbed into the bloodstream and then go to the peripheral cells. So what happens once a monosaccharide gets into the cell? Stage 1, digestion. Got the monosaccharides. We're going to focus primarily on glucose. So the very first thing that happens when glucose enters a cell is it gets phosphorylated. So an ATP is used to stick a phosphate onto glucose and you get glucose 6-phosphate. We'll look at these reactions in more detail later, but this does a couple important things. One, it activates glucose, and second, now that glucose has this phosphate attached to it, it cannot exit the cell, so this glucose is now stuck inside the cell. For most cells. There are some cells we can talk about later when we get into the Cori cycle where that phosphate can be removed and the glucose can leave, but for the most part, for most cells, when glucose gets phosphorylated, it's owned by that cell. So then glucose gets broken down through a number of steps, and it only shows 3 arrows here, but there are actually 10 steps where glucose is broken down into pyruvate. Now, one way I would recommend remembering these reactions is to keep track of the number of carbons. Glucose, as you recall, has 6 carbons. Pyruvate has only 3. Pyruvate or pyruvic acid, same thing. Pyruvate is a conjugate base of pyruvic acid. So 6 carbons gets broken down to 3, but you notice there's a coefficient of 2 here, so 2 C3 units. This pyruvate then can be converted into a number of different things, primarily into acetyl-CoA. We talked about coenzyme A in the previous lecture, and we'll see where this goes into stage 3, then the common pathway. In any event, it requires some input of ATP, and we get some ATP out. So if you look at there's a net investment of 2 ATP at the start, and we get 4 ATP out. So the yield, it takes 2 ATP to activate it, and then we get 4 ATP out. So the net, right, is 2 ATP come out of glycolysis. It's really not a whole lot of ATP. So the first, I mentioned there's 10 reactions. The first 5 they refer to as energy investment, and this first reaction, you should know, is catalyzed by hexokinase, very important enzyme. I think we talked about hexokinase in the lecture on enzymes and turnover number and conformational changes and stuff like that. It's a really kind of a neat enzyme. And it will stick phosphate onto the carbon number 6 of glucose, so we get glucose 6-phosphate. Then there's an isomerization reaction where glucose is converted to fructose. You can see why we're kind of focusing on glucose, because fructose, one of the other monosaccharides, can enter here at this stage with fructose 6-phosphate. So hexokinase, again, catalyzes this reaction and essentially, for the most part, for most cells, traps glucose in the cell once it's phosphorylated as glucose 6-phosphate. So we can say here, for most cells, phosphorylation traps the glucose into the cell. There's no way for that cell to export glucose out of the cell once it's phosphorylated. There are a few cell types that do have a dephosphorylase enzyme which will remove this, but this reaction actually is irreversible. And I should mention that again. This is irreversible. Under normal cellular conditions, this reaction is only one way, irreversible. You can't go back from glucose 6-phosphate to glucose. So there's an isomerization reaction, makes fructose 6-phosphate, and then it gets phosphorylated again. So it gets phosphorylated on carbon number 1 and carbon number 6, carbon number 1. So here we'll say this is 6, this is 1. And then the next step, it gets ripped in half into 2 3-carbon units. So 6 carbons go into at the top, and 2 C3 units come out. And that's the first 5 reactions. Something else I should mention, this first step, hexokinase, this third step is catalyzed by phosphofructokinase, The names for these enzymes are on a slide coming up. But this step is important. This is also irreversible. It's an irreversible step. Many of these steps that involve ATP are irreversible, and it's also known as the committed step. And we'll talk about why it's called the committed step later. So that's this third step. The next step, where this fructose 1,6-bisphosphate is cleaved, giving us two of these guys. Now, we refer to this as bisphosphate, not diphosphate. If you remember, a diphosphate would have two phosphates joined in a, what was that kind of bond called? What was that? Important high energy bond. That was a acid hydride bond. This is a diphosphate. And to distinguish a diphosphate, which has this anhydride bond, to simply a molecule that has two phosphate on it, we use the word bis. So bis means two. So that's the energy investing stage. You notice we use 2 ATP. Glucose comes in. 2 C3 units come out, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. A lot of the names here you don't need to, I'm not going to ask you to memorize. You should know hexokinase, of course glucose, but the individual intermediate names are not all that important. So here they're showing you actually the first step here, hexokinase. This is carbon number 6. Right. This is a phosphoester bond. Right. An acid combines with an alcohol to give us an ester, phosphoester bond. And you'll notice this reaction, this arrow is just one way. Irreversible. Hexokinase is irreversible. The next step, glucose 6-phosphate to fructose 6-phosphate, the arrows, there's 2 arrows, up and down. So arrows like that. Like that. So this is reversible. Does not involve ATP. This third step, this committed step I talked about, again, irreversible. This is going to be important later. It's irreversible and involves ATP. Cleavage of these bonds here, remember, 7.3 kcals per mole involved in this. So some energy is used to activate this molecule, and then it's ripped in half. Now, once you break this in half, you get 2 3-carbon units that are different. Okay, and it is only the glyceraldehyde 3-phosphate that goes on to, they don't have the name here, but this is glyceraldehyde 3-phosphate. This is glyceraldehyde 3-phosphate. These go on to the remainder. Dihydroxyacetone phosphate does not, but there's an enzyme that will convert this to glyceraldehyde 3-phosphate. So both 3-carbon units end up going on to the rest of the reactions in this series. And again, this reaction is reversible. It goes back and forth. No ATP is used, and it's reversible. Where ATP is used, you see just one arrow, and it's irreversible. The next reactions, 6 through 10, are the energy-producing reactions. So we've ripped this guy in half, 2 C3 units. It's the glyceraldehyde 3-phosphate that goes down. And now we see our NAD. So this is an oxidation. Right? NAD is reduced to NADH, and glyceraldehyde is oxidized to glycerate. This is an aldehyde. Glycerate, that's an acid. Glyceraldehyde, clearly an aldehyde. Glycerate is an acid. So this thing is oxidized. It goes from the aldehyde to the acid. And it's NAD that's used to carry out that reaction. In addition, this is also what they call substrate level phosphorylation. If you notice on glyceraldehyde, there's this pink guy here, that's the circle, that's the phosphate. Now you get to 1,3-bisphosphoglycerate, so now there's 2 of them. The 2 here is simply a consequence of the fact that there's 2 C3 units coming through. Likewise with the 2 NADs, 2 NADHs. So everything's going to be multiplied by 2 now because we have 2. There are 2 C3 units, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These 2 C3 units come through, so now we're multiplied by 2. Alright, so we go from the aldehyde to the acid. It's an oxidation. And then ADP is phosphorylated. So this is, again, substrate level phosphorylation. The energy in this, in one of these, in this, let's say it's this one here, this bond, the energy came from this oxidation. Right, so just when you burn, like when you burn any fuel source, you get light and heat, you get energy from it. We're doing a similar sort of thing, except we're not burning it in a flame, we're burning it, oxidizing it with these coenzymes. Then energy is stored here in this bond, and then it is transferred to ATP. So there's, again, everything's multiplied by 2, so 2 ATP here, and a similar sort of thing happens at the end, 2 ATP, so we get 4 ATP, and this molecule pyruvate. Let's look at the structures now. Okay, so glyceraldehyde 3-phosphate. This is the aldehyde. This is not an aldehyde. This is a phosphoester. This is still a phosphoester. This now is an acid anhydride. Right? This is a phosphoric acid and the glycerate. So this is an acid anhydride. This is a high energy bond here. Whoa, what the heck happened there? I don't know what happened here, but this is an acid anhydride bond, and it's high energy, right? That energy then is used to phosphorylate ADP to ATP, so now it's gone. That phosphate here on the top is gone. This step here, also reversible. This step here, also reversible. All these guys are reversible, except for this last step. So what happens here is, again, the phosphate is moved to the second carbon. The alcohol here gets converted to an enol. So this is an enol. The reason it's an enol, this is a double bond, phosphoenol. This here is a double bond. Maybe you can write that a little bit clearer. There's a double bond here. And this carbon oxygen phosphate, this is still a phosphate ester, but now because it's an enol, this would be the ene, the ol would be the CHO if there wasn't a phosphate here. This is equivalent to equivalent to essentially an acid. This carbon has been oxidized. The last step then, the energy in this bond is used again to phosphorylate ADP to ATP, and we end up with pyruvate. Let me go ahead and draw pyruvate in a little bit different way. Okay. So that would be propanoic acid, 1, 2, 3 carbons. All we've done here with pyruvate is we've put a ketone on that alpha carbon. So that's propanoic acid. What does pyruvate look like? It's pretty much the same thing, except we have a carbonyl, a ketone, on that alpha carbon, that second carbon. So there's pyruvate. So the difference here on that second carbon. All these reactions are reversible, and they involve 2 substrate level phosphorylations. The reason we call it substrate level is because the substrate is ADP, it gets phosphorylated to ATP. So again, and you should multiply each one of these through by 2. So there's 2 here, so a total of 4 ATP. Right. The net result here is glucose gets converted to pyruvate, and we get a net of 2 ATP, and significantly also 2 NADH. ATP of course is high energy, but some of the energy involved in the oxidation of glucose to pyruvate is also stored as NADH. Unfortunately for the cell can't really use NADH as energy currency like it can for ATP. So we'll see later how this NADH gets converted to something more usable like ATP. So I made a point of talking about which enzymatic steps were reversible and which ones weren't. And the reason for that is the irreversible ones, those are the ones that are going to be used for regulation. So the first step, number 1, hexokinase, is going to be regulated. It's irreversible. Reaction number 3, this, if you remember, this was the committed step. And reaction 10, the last step. So if you think about it, we're going to regulate the first step where glucose enters glycolysis pathway. We're going to regulate the last step where, not really glucose anymore, but where the pyruvate leaves. And then also this third step. I'll talk a little bit about why that is. So when it comes to regulation, there's something called the energy state of the cell. When the energy state of the cell is low, cell says, hey, you know, I need to get more energy, so I'm going to go through catabolic processes that produce energy. Let's just talk a little bit about regulation in general. We'll call this a high energy state on the top and low energy state. Okay. So when the energy state of the cell is low, you're going to go through catabolic processes to increase the energy. When the energy state of the cell is high, you can use that energy to do things like running around or thinking or making new proteins or growing. You're going to go through anabolic processes. Glycolysis is a catabolic process. It produces ATP, right? You say here, make ATP. Anabolic processes use ATP or consume ATP. So when would the cell decide to, and by the way, these are all enzymes that are regulated. They're allosteric enzymes. Remember that term, allosteric enzymes, by 3 allosteric enzymes. So hexokinase is inhibited by high levels of glucose 6-phosphate. This is a type of feedback inhibition that we talked about in the chapter on enzymes. So if we already have a lot of glucose 6-phosphate, we don't need to make more. So we're going to slow down hexokinase. So if hexokinase is inhibited, that means any glucose gets into the cell, doesn't get phosphorylated right away, and it can leave and go do something else. All right, so this prevents glucose from being trapped in the cell. It's inhibited by high levels of glucose 6-phosphate. So the glucose 6-phosphate essentially signals high energy charge or high energy state of the cell. For reaction number 3, this is inhibited by high levels of ATP. Well, again, if we have high levels of ATP, this signals a high energy charge of the cell. Signals a high energy state of the cell. And if we have high levels, we don't need to make more, so it's going to be inhibited, this third step of glycolysis. Conversely, it's going to be activated when levels of ADP and AMP are high. Right? High levels of ADP and AMP. These high levels signal a low energy state or low energy charge. When the energy is low, what happens? Well, we have to go through some catabolic processes and make more ATP, right? So this step is going to be activated when energy levels in the cell are low. The whole point of glycolysis is to make ATP. So another allosteric enzyme, this guy's going to be inhibited by high levels of ATP or acetyl-CoA. Acetyl-CoA isn't really part of glycolysis. It's actually the one step down the road. But when you have high levels of acetyl-CoA, this also signals high energy state. We can talk more about that later. So glycolysis is regulated at 3 steps, 3 irreversible allosteric enzymes. All 3 of them involve ATP. The first 2, or I should say the first reaction and the third reaction, these are in the enzyme or the energy investment stage, the first part of glycolysis, and then the last reaction when the carbons of glucose leave the pathway in the form of pyruvate. 3 regulated by 3 allosteric and I should say irreversible enzymes. I'll let you think about this question. You can pause it if you like, and the answer's here. In the next video, we'll talk about what happens to those carbons of glucose after they've been converted to pyruvate.